All right, good evening and welcome to Out of This World Science, Space Health and Space Biology Symposium. And this is a really great educational program that's been co-hosted by UC Space Health and also by NASA Biologic and Physical Sciences. So it's a great opportunity for us. We have people in person and we have quite a few people on Zoom. Sorry for the Zoom people, but in-person people, there's food and beverage outside, so help yourself. And I hope whoever's on Zoom has their own nourishment going. My name is Einor Sawyer, and I direct the Space Health Program here. I'm an orthopedist. I'm also involved in health tech innovation and innovation ventures, and always happy to tag up with anybody super passionate about studying space research and development and space health in particular, because I like the concept of space and all of the exciting principles of it, but I also really care about what we can learn and benefit from by doing that research and bring it back to Earth, particularly applied to terrestrial healthcare. So I have a term I use. If anyone's asking you, why are you interested in space? Why are you spending time when we have so many problems on Earth? Tell them it's for the ROE instead of the ROI. You're investing your time and your knowledge for the return on Earth. So you may use that, and it's been motivating quite a few people to understand why we're passionate about what we do. Okay, let's move on. I want to do a couple of thank yous to this room here in this audience. The agenda you've seen already, you signed up for it. We are very fortunate to have Dr. Walter Alvarado and Dr. Sergio Baronzini with us tonight. We'll get to their intros in just a second. And the meeting portion will go till about 7:30, and of course we can network afterwards. Part of that will be Q&A, of course, so interactive, and people on Zoom can push their questions, and we'll be gathering those as well. I want to give a big thank you to Dr. Sylvan Costas, who's joining us remotely tonight, and he heads up the Open Science Data Repository and many other aspects of space biology and health research down at NASA Ames. And he's been a great collaborator with me, and I've learned much from him over the years, and we'll continue that, I think, Sylvan. And then I want to thank Lauren Sanders, who also is from NASA and with Sylvan's group, who's really helped me get this stood up from a logistics standpoint, and her colleague Lavorka as well. And all of these people are way more than meeting organizers, but they've been kind enough to lend their time to me in that capacity. So we're very lucky with the people we get to work with. So kicking over to the agenda, I'm going to start out by introducing the concept of these meetings. We want each time to have speakers from NASA and speakers from UC Space Health, UCSF primarily, but also the UC system. So we have had speakers from Berkeley as well, and we will continue to do that. The idea is have representation from both sides and some intersection on the topics. May not always be, but you might be interested in the prior sessions, and all of the sessions are recorded, so you can go online and see the links to them. But we've had some fascinating presentations prior to this. But let's dive in tonight and start with Dr. Alvarado. And we're really fortunate to have him come all the way up here to visit with us. And he is a biophysicist in Space Biosciences Research Branch at NASA Ames Research Center. His research experience involves designing machine learning models based on physical models to study complex biological mechanisms. And his current research interest is focused on identifying biomarkers indicative of space radiation, and we have some space radiation experts in the audience. Fortunate to have people gather around that topic. And then aiming to improve real-time health monitoring and develop protective strategies for astronauts on deep space missions. He also serves as data scientist to both the Open Science Data Repository and the NASA Biological Institutional Scientific Collection. And in these roles, he applies his machine learning expertise to improve the data curation process and to help increase the access to and the understanding of important space biology data. And we have teams here at UCSF, Dr. Barrios, who's in the audience with us today, who have already been working quite a bit on intersecting the space radiation research with, of course, our human care and radiation oncology here at UCSF. So I would like to have Dr. Alvarado come up and begin. His talk is titled AI Curation Methods for Scientific Data. Each time we'll have a computer switch, so you'll see that happening, but it's quick.
Thank you for the introduction. Hi, thank you. I can use either one, right? Okay. Well, thank you so much for being here today. I really appreciate it. I'm really excited to share some of the work that we've been doing at NASA Ames. My name is Walter. I am a researcher with the Space Bio Research Branch at NASA Ames. I also serve as the AI and data scientist for the NASA Open Science Data Repository. This talk is about AI curation methods for scientific data. So AI for curation, but it could have easily have been data curation for AI. Because the irony is that a lot of these AI models wouldn't work without curated data in the first place. And so I feel like that interdependence really highlights the importance of having really strong data foundations. So a little bit about the work that we do at the space biology research, which falls under the biological and physical sciences. Our team tries to study spaceflight and radiation and how to fix living organisms by studying them either in space or ground analogs. The goal is to understand how factors like microgravity, radiation affects these living systems. Here we have one of our NASA scientists, Butch Wilmore, setting up the first rodent research project, the hardware, to ensure to see if it's safe and if these experiments can be conducted in the International Space Station, and also studying muscle atrophy in space. After these experiments are conducted, these tissues are stored in our biobank, what we call AMBISC. And the data that gets collected from these research experiments gets ingested into GeneLab, a data system that collects a lot of these experiments and makes it accessible to the rest of the scientific community. Ensuring that it's open and discoverable for other people to be able to do their experiments based on these really rare experiments. Now, a lot of these projects can be sort of are led by different groups at NASA Ames. We have LSDA, which stands for the Ames Life Sciences Data Archive, which is responsible for non-human physiological, phenotypic, and imaging data. We have GeneLab, which curates omics data, and where users can go upload their data, share it, store it, analyze it, and sort of study different, download a lot of these different studies and experiments. And of course, NBISC, the NASA Biological Institutional Scientific Collection. NASA really loves its acronyms. So it's a biorepository of non-human samples from experiments that are NASA funded and that investigate different types of stressors that can occur in space. And the idea is to make this data accessible to everybody by ensuring the data is something that's what we call fair, maximally fair. To sort of take a look at the type of data that we have in our repository across the GeneLab and LSDA, we have around 500 studies, almost 1,000 different data sets, looking at almost 45 different species. And across collaborations with the European Space Agency, JAXA, and that adds up to like 160 terabytes of data that needs to be curated and accessible for everybody else. And so you can see that that's quite a tall task. Just like every other sort of data-centric businesses and organizations, we run into a lot of the same issues that they're running into. Things like data silos, right? You have data that sits in two different locations within the same organization, and that makes it very difficult to share. And of course, scientific data is very specialized, very complex. So it makes it that's added complexity on top of all this redundancy, which I know I'm very much guilty of. It's a terrible habit I picked up in grad school where I would make copies of my data because I was afraid I was going to lose it. And instead of deleting it, I would just make more copies. And then obviously incompatibility. Once you have access to this data, making sure that it fits within the pipeline that you have implemented into your organization. And so to address these issues, like any good self-help book, you find some really good principles and you align yourself to them. And so here we sort of adopted the FAIR data principles, findable, accessible, interoperable, and reusable. When we say findable, what we're saying is ensuring that the data is accessible, not just to humans, but to machines as well. The analogy I heard once was that you wouldn't open up a restaurant, send out different trucks for different ingredients, and wait for those trucks to come back to make your meals. You would essentially have that data ready to go and make it accessible to everybody else in the team. And that accessibility also requires it to have security and making sure that the people who need access to that data are the only people that need access to that data. But if the data can be open and free, ensuring that that is something that we strive for. And reusability, ensuring that the data is also ready for not just for AI applications, but also trying to see if we can sort of be the basis for future research. We're all familiar what happens when data is not fair. I would like to thank my colleague, Dr. James Costelletta, for sending me some examples of issues when he's running his experiments. And I mean, we're all sort of familiar with it, right? You're trying to extract some data, and maybe there's like a trailing space at the end of that string. And so you spend a few hours trying to figure out why you can't extract the information. So things like that can be really easily solved with some programmatic methods. But there are different times when you'll receive a data set from a different PI, and we all know that data sets come in all different shapes and sizes. And the data that you're looking at might be exactly what you're looking for, but might have different vocabulary, but have the same semantic meaning. And that's really where the power of large language models really come into play and really help us start to standardize and curate a lot of this data set. Without going too technical on how large language models work, I think we're all sort of familiar with them. ChatGPT is sort of the really popular one. It's a transformer model. So you have a couple of strings, you feed it into the model, and it can approximate the next word, the next token. And it does really great with natural processing data. But we can leverage these large language models to allow us to do a lot of the data curation side and really minimize the amount of energy and time that we spent curating scientific biological data. And so I'll go over some few examples and some of the methods that we've implemented. A lot of this has been done with a lot of effort and great detail by our interns, our scientists, project managers, and a group of hundreds of volunteers that go through our working groups who give us feedback on developing these tools. And some of these tools sort of apply throughout the whole curation process. At the beginning, like automating the data alignment process, metadata tagging once your data has already been ingested into the repository, and even some really cool things like generating graphical abstracts from these experiments using these large language models. And of course, everyone's favorite, a chatbot that can retrieve and get the information that you need and give you more context about the knowledge database that you have in your systems. So like I mentioned earlier, you can apply this throughout the whole curation pipeline, and that's just a fancy way of saying that you can use Gen AI for a lot of different things. One of the first use cases that we tackled was trying to align the data that we get from different investigators. A lot of this data comes in different formats, and some of these validations can be done pretty quickly, really easily with really programmatic ways. What I tell my students when they're developing these tools, even though it sounds a little counterintuitive, I always say, like, try to stay away from AI for as long as you can. And the analogy is you have a locked door, and if you just look around, you'll find a key. And that key will open the door 100% of the time, and it doesn't make a mess. But sometimes there isn't a key, and it's okay to drive a tank through that door. And that's what Gen AI is, right? And so, but combining these two approaches, a programmatic approach and these new tools with generative AI, you can build some really robust tools that allow you to align your data and spend less time trying to worry about how to format the experiment and focus more on the actual science. Here's sort of a really easy example. So we have a data set and we have something that we like it aligned it to. So here on the left-hand column, we have the fields that we need filled out, the metadata that we need to extract from whatever particular experiment. And you can see that on the second column, that is the data that gets ingested from a particular PI. And, sometimes you can have things like that aren't supposed to be there, right? Like these little extra characters, so on and so forth. And you can use things like, again, like a Levenshtein distance, which is just a similarity metric between two different strings to tell you how similar these two words are, or how similar these two lengths of string words are. You can set a threshold and then use that as a validation tag. And that really does a lot of the heavy lifting. That's essentially the engine that can do a lot of the matching. But once in a while, you'll have instances where maybe the data that was submitted isn't using that particular vocabulary, but it's using the same semantic meaning. And that's really where a large language model can come in handy. And so we built these tools to allow us to find these matches automatically and generate these new data sets that we can then ingest into our repositories. Now, I'm not going to go a lot into the code, but the way you kind of do this is that you impose a schema to the output of the large language model. So you can ask models, hey, there's a list of things, based on the context, which of these could be the best match. And you can format it in such a way that the output will come out in some like JSON formatted way. And then use tools like Pydantic, for example, that will verify that the output that comes out is following this particular schema that you're interested in extracting from it. And if it doesn't match it, that's fine, because you can just use it as a validation check to say, all right, well, we can just move on, and maybe a data curator can help us with that later. Now, these tools are great, but we obviously know that not everyone knows how to code. And so it's useful to develop tools that everyone can use. And then so over the summer, I had a great software engineering intern, Kayvon Crenshaw, who developed this GUI that allowed us to actually implement it and allow other people to be able to use it. And it's great because you can sort of, I can see, I think I have a video. Yeah, and it's great because you sort of select the template that you need. So in this case, MBIS intake form. You can then upload the data set that you need, and then it will try to find the matches using things like the Laverstein distance. And if it doesn't find it, then you can start to enable it, and you can check the box and say, hey, I need some AI-enabled matching to be done here. And then it'll think, populate some answers. And in the back end, everything is being sort of validated, verified, and ensuring that the matches make sense. And then once it matches, you can see that you can kind of have these things filled in properly. And then you can export your file, and then we can use it to ingest it into our repository. So that just saves a bunch of time. And that's what's so great about some of these tools, is that it's saving us time and energy to focus on the science. But using the same types of approaches, we can also implement it and use it for things like generated metadata tagging. So once we have the actual studies in our repository, how do you go back and how do you start to add additional fields that you might have missed? A lot of this stuff at first glance isn't necessary, because you think to yourself, well, I'm reading a study about astronauts, so I know there's astronauts in this study. But as we start to use data to train these AI models, it's nice to have a way to extract that information using metadata tags. And so you can go back and try to say, okay, well, how can I sort of append to the structure or the projects that we already have in our repository? And so one of our developers, who's actually here today, sort of uses this same approach to be able to go back and apply these metadata tables or metadata tags to the projects to make the data more robust. And that gives us a liberty and more freedom to sort of explore the data in different ways by adding these particular tags that weren't there. You ask the large language model, hey, based on the context of these studies, can you add or assign these metadata tags? And then use that to sort of build these really cool graphical metric websites to explore what's on there. Does anybody have any questions before I move on? Well, cool, awesome. All right, so that's at the beginning process of sort of this data curation process. So the next step, or the next kind of tool that one of our interns, Harlan Phillips, built over the summer was this graphical abstract generator. Even for seasoned scientists, it can be really difficult to sort of make sense to the experimental protocols that were developed, and visualizing these experiments can really help. And so he took the time to build this tool that allows researchers to essentially take this study that doesn't have any type of visualizations into it, extract the information from that study, right? So all this freehand text, submit it into this portal, and then it can generate tabulated data, but then it can also, if you wait a few seconds, a graphical abstract. So it gives you an idea of what these experiments were all about, the types of radiation they were exposed to, how long they were exposed to for, the types of tissues that were collected, the subjects that were looked at, by essentially combining programmatic methods with AI, which is really a cool thing to watch. Okay, and so on to everyone's favorite use case, chatbots. So chatbots are great because it allows you to discover and visualize discover data that you might have in your repository. And it does that by trying to find information in your knowledge database, and use that as context to give you answers based on the query that you have. So here we have our little chatbot. You ask it, hey, any studies that looked at bone density loss in spaceflight, and it can point you directly to a particular study. And then you can ask questions about it, like what type of assays were conducted, and then have it summarize and give you more information. And the nice thing about these methods is that you can fine tune them, and then you can say, all right, well, maybe extract more than just one study, and then summarize it and give it different types of prompts that will be great for your particular use case. And then the other thing great is that you can implement it into your open science data repository or into your own repository, and then have it sort of be this extra seamless tool that you have in your system that can be added benefit of being able to serve as like a search bar for different types of for different types of queries. And the magic behind all this is something called retrieval augmented generation. So a lot of these large language models are trained on data that's publicly open available, but not all data is available. Some of that data is behind, it's private, so you can't really use that to answer the question. So if you ask the chatbot, hey, tell me something about this particular study, and that data wasn't in its training set, it's not going to give you an answer. And so one thing you can do is you can create these databases that have the information about your repository and then use that as context. So then when you start to ask the large language model a question, it can say, okay, well, I'm going to look for the right document and then use that as context for the answer that it provides in the end. So that's one approach. There's other approaches that you can apply to your RAG stack, is what we call this, like knowledge graphs. I think Sergio is going to talk about later right after this. And so that's kind of the sort of the magic sauce behind these chatbots, especially for these particular use cases. And so, I mean, in summary, I think it's pretty obvious that a lot of this data curation stuff is important, not just necessarily for training AI models, but it's also important to maximize the amount of output that you get from these really rare experiments. Not every day that we get to go and do experiments in space. And so by making this data open, accessible, and easy to read, we open that possibility to other people to be able to go in there and do their own particular types of studies. So for fun, I asked the Dolly, what do you think the future of AI curation and AI looks like? And it's pretty terrifying. You can see it's the humans are working, and I don't know what that is. So anyways, any questions? Perfect. Thank you. Yeah.
I don't know what people think, because I know they will too.
For sure.
We're going to do a few questions now for Walter, and then he speak and then we'll do stuff. Why not?
We're definitely going to. I think it's because that is our number one fear. I think that, especially when you're working with scientific data, like it's important to be accurate. It's important to have the right values. And I think the number one fear is that, you have these large language models doing automatic things, and then, it's processing hundreds of thousands of data sets. And it's just not practical for a human to go in there and look at everything. And so building tools that prevent that, I think, is like the number one priority, specifically for scientific data. So yeah, that's definitely my fear. Yeah, for sure.
Honest about that.
Yeah, no, of course.
And a couple questions. Yeah. So at this point, can you give us a sense of how much use the system gets from outside of NASA?
Yeah, definitely. A lot of these tools and these processes are being implemented.
And that's kind of what we're doing, is that we're seeing what's out there and seeing how that can apply and how that works for us. A lot of these, specifically in NASA, we are seeing different divisions like from astrobiology to heliophysics, adopting these methods. The data standardization is sort of like the big one, ensuring, because we have so much data. And so it's important to make sure that that data can be curated, it's accurate, and these large language models allow us to do that. These tools are being used everywhere, and some of these methods are being developed specifically for these types of use cases. And so I know, for example, our tool, the data alignment tool, we're using it for the experiments coming in, or the samples that we're getting from different experimentalists. And that really cuts down on the time of trying to manually find semantically things that were there before, right? So yeah, it really does.
You know, big problem. Yeah. The big problem, it's great how much, this has really moved forward in the last 5 years, right? I'm wondering, has it moved far enough out that other people come from outside of NASA come in and use the data in ways to inspire experiments or yeah. So you got it all, but are people now that you
Yeah.
have people come?
Yeah.
You say, speak this for Zoom on the same.
Yeah, that is what I mean is that like people
Using the open science data repository for their own type of research? And the answer is absolutely. Yeah, there's a huge return on investment on a lot of these studies. So these studies have been published. We republish them with the open data accessible to the rest of the community, and we've seen people publish on top of that and look at that samples. We have people who go in, request our samples, request this data, and do follow-up experiments on all either the samples and use that data for their own type of analysis. So we definitely have had a fair amount of people being able to utilize the open science data repository. Now, the tools themselves, they're pretty new. And so a lot of it requires, making it accessible to people who don't know how to code. And so those things are definitely new. But as the field matures, we're definitely getting to the point where anyone can pick it up and start using it. So, yeah.
Great talk. And more than a question, I have a comment and then praise for you, because it's a great idea to use those LLMs to curate data sets and find matches. So the matching tool would show it's fantastic. It's good news for so many different things. Again, another example of any type of experiment that produces data, you need a human to get to see what does it really mean in terms of species, whether people have less or extra space. So I think it's a great application of all these. So this kind of nuances, slow language stuff.
Yeah, I mean, I think when you're doing a lot of the science, it's having to say, okay, this needs to be formatted, this needs to be curated. It can take days, and it takes energy, and it takes time, and it takes resources, it takes labor, which is expensive. And so having tools that can minimize that and ensure that we're just focusing on okay, what was the experiment? What did we find? What was the importance of the experiment? And how do we make other people know that this experiment exists? Experiments are very rare. And so having people be aware and know that these things exist is a valuable tool, I think, for anybody that's interested in space biology. So questions?
Oh, they're not here. Do you want me to switch some better? Oh, sorry. Oh, God, sorry. Oh, no, it's okay.
So these tools that you showed us, the AI visualizer and stuff, is it openly available, or do we just Google?
Yeah, no, they're available. So you can go on the GitHub, and I should have posted that up there, but we have a GitHub where a lot of these tools are accessible. And it's great because it's open, people can use it, and then retrofit it to their own use case. So if you have data that you'd like to align, the methodology is there, and you can look at it and have somebody else kind of take care of it and format it to your need cases. Yeah.
Thank you. Thank you so much for your talk. Excellent talk. I just have a question about the metadata generation using AI.
Yeah.
I think that's a very, very interesting application of large language models. But my question is, while this tool is getting created, how do you benchmark the performance of the metadata generation using LLMs? Especially when you said that some of those studies are by design rare and hard to find, and probably not something that the AI models are originally trained on, or at least not even close to what the AI models were trained on. How do you make sure that the AI models are doing the correct job? How do you generate a benchmark to see what type of studies do AI label it better, or what type of studies do AI not really do a good job? Stuff like that. How do you make sure?
Yeah, there are tools out there that are being designed, and I think the important thing to remember is that these tools are new, and a lot of the benchmarking tools that benchmark these new tools are also new. So you have new tools, new benchmarking tools, and so right now the way that we're working with this is using easy cases, like like just simple binary questions like true, false, and then being able to map that, graph it, and then see if it aligns with what we have done. We have a great team of curators that to know these experiments and know the actual what these things should be, the ratios and stuff. And so we compare it with actual scientists and curators who know these answers. And the hope is that as these tools get better, because they're not good. I mean, they're not perfect. I meant to say they're good, but no, they're not perfect. And so there are definitely instances or cases where it doesn't or misses the mark. And I think it's important to make sure that we have a tool set that allows us to capture that information. So that's kind of where we're at right now. Right now, good. I would say medium. Yeah, I can't give you like an actual quantitative value, but they're pretty good. Yeah. So yeah, question.
Hi, yeah. I had a question about the retrieval augmented generation that you were
Yeah.
talking about. I was curious if you ask a question that's outside of the context window, as opposed to like a normal LLM, which might hallucinate. Does this also hallucinate, or does it simply say doesn't fall within the context window, or does it kind of try to impute the data, or how does it react?
So that's actually why we implement RAG in the first place, is to minimize the hallucination. So when you ask it a question, right, and if it didn't have any context, it would just hallucinate and say, oh yeah, this is what this is, right?
Yeah.
And it would not the answer. And so RAG minimizes that and ensures it doesn't occur. There's other tools you can also implement on top of RAG that can minimize the hallucinations. And that really helps if you have data that's already really well curated, because then you can have things and tools or functions that can go into the query. So let's say you're talking to a large language model and you ask it a particular question, like look up this particular study, OSD 488, right? There's a keyword in there, and that keyword can be then used to retrieve a specific document relating to that. So it provides even more context to reduce the hallucinations that occur. And so there's different methods and methodologies that are coming out. And so, playing around with these is sort of most of the fun. Yeah.
Thank you.
Yeah, of course.
All right, very good discussion. We have a question from Zoom, and this is going to be a warning to you, Walter. It's from Sylvan Costas.
Oh no.
This should be very interesting. Are we able to pipe Sylvan into the room? Great. Hi, Sylvan, you're on.
Oh, sorry. I can you hear me? I lower my hand.
Yes,
Yeah.
perfectly.
Okay. Well, I guess it was not really a question, but more a comment, because I think I heard a question about how do you use the AI to really make new discoveries, and what's interesting in Walter's work is that it's really what he's focusing on right now is using AI to fill this metadata schema. So OSDR has built those very stringent metadata structure. And the reason we're doing this is that we want to control the vocabulary, we want to the information, so that what happens is that we call that AI readiness. That means that the database is AI ready. So what's interesting here in this exercise is using AI to fill up the metadata structure. And the better the metadata structure, the more AI ready the database itself is. So, and I think this is going to be the future. You're going to see these kind of like a Pandora box, where you open a box and inside that box there's another AI, and you open the other box and you get another AI. And so what's going to happen with OSDR is we're going to have the power of AI to fill the information that will then be used by other AI, because once we're AI ready, we can really start to answer the question that was asked by someone in the audience, in terms of what do we discover with this. The only way you can bring multiple studies together and have that bigger picture is when we really have this very accurate representation of the data in a way that is unambiguous. And that's what we're trying to do with this database. So I think the future is going to be very interesting. The more we curate the data the way we're doing it, the more powerful the database itself is going to be. So I wanted to make this point.
Yeah, thanks, Salon. Also, I saw Ryan on the call up there, and another good point is that when we're matching and trying to find the right ontologies, that can also be a big discussion, not just for AI, but for humans. Like I've been to those meetings, and those ontology meetings can go for a very long time. because it's like, no, it falls under this. And it's like, and so having the power of like our working groups and everybody that works at NASA, it really does help us sort of come with have a solution standards that we can use to implement. So the hope is that when we do use these data sets for training other AI models, that there will be a lot more accurate. So
that last point you made is a really important one for us in healthcare as well. The ontologies and building the ontologies and also intersecting them and getting consensus from the different groups. So we may be borrowing some of what you're doing.
Perfect. Yeah.
All right. Thank you so
Thank you.
much. Let's give another round of applause for Walter. That was fantastic. So now we have the privilege of hearing from Dr. Sergio Baronzini, who is here at UCSF with us, but he also does quite a bit of work with NASA, and you just saw one of his collaborators on screen, Dr. Costas, and I have the good fortune of also being one of his collaborators here as a colleague at UCSF. And he's going to be speaking on the Spoke Biomedical Knowledge Graph, applications on Earth and in space. Dr. Baronzini is a distinguished professor of neurology here at UCSF. He's also a member of the graduate program in bioinformatics, the Institute for Human Genetics, the Bakar Computational Health Sciences Institute, ImmunoX, and the California Institute for Quantitative Biology, or QB3. He holds the Heydrichs Friends and Family Endowed Chair in Neurology, and he earned his degree in biochemistry and a PhD in human molecular genetics from the University of Buenos Aires in Argentina. He's going to tell me how to say that better. Dr. Baronzini moved to UCSF, and he now specializes in the analysis of complex hereditary diseases, with efforts focused on MS, but other disease states too, including different types of immune disease and also cancers. And he's porting his model and his expertise looking at risk prediction, prevention, et cetera, across different disease states, and also looking at how this can benefit work that's being done in space health and leverage space data to advance what we're doing here on Earth. So I'm going to turn this over to you and let you also do a further introduction of yourself if you wish, points you want to highlight, because his bio is several pages long.
Sorry about that. So no, it was great actually. Thank you so much, Einar, for the opportunity to share some of what we've been doing in this space, pun intended. So I also used DALL-E to create this image, which I think it was pretty good, just to put together knowledge graphs with space exploration. And the point that I want to discuss today and share is our work on knowledge graphs, and particularly in the Spoke knowledge graph, which is a biomedical knowledge graph, and how can we use that to inform the work that you guys are doing at NASA, but also some of the analogs that we can find in the medical world and see how we can cross-pollinate. So this is another DALL-E generated image, and this is something that I found, I think this is a great example of how this things work. The prompt that I gave to this one is create an image that can represent the singularity that occurred in 2017, in which 2017 was the year in which there was as much data generated in that year and in the history of humanity. So from then on, we've been generating yearly more data than what we have been able to accumulate from 10,000 years ago. So, and this is quite an interesting representation of that. So now, 90% of the data that we consume today was generated 2 years ago. We're in the exabyte scale of data generation per year. We're about 35 exabytes for 2024. So what do we do? One of the big problems and one of the big challenges that I think is in medicine is that of data integration. How do we bring together all of the different modalities that we can characterize a patient on? And of course, each researcher and each doctor will prefer their favorite modality to be the one that solves the disease. But in reality, humans, patients are multidimensional. And sometimes we will need a simple clinical evaluation,
to solve the problem. Sometimes we'll need a lab test, and sometimes genetics will do it. But sometimes, and I would argue a lot of times, none of those. And perhaps it is a way to infer the condition of a patient by means of being able to integrate. And we can integrate in different ways. We can integrate by creating a statistical model and ask the model, okay, these are all my variables, this is multivariate model, how well can this explain the phenotype? But that is a statistical model that does not take into account biology or biomedicine at all. So the approach that we would take is that of a real integration. That's why we chose to go into knowledge graphs. And let me just give you this representation that I like about the hierarchical organization of biological complexity that we most will probably agree that we start at the bottom with DNA, the genome or the metagenome now. Then you have the next layer up might be gene expression, which could be interpreted as the most immediate phenotype, if you wish. And then you have proteins above that. And proteins interact, and then pathways emerge as part of the protein interaction. And then you have cells, and cells are great integrators of information, and they can produce certain outputs. And then you have organs and tissues, and all the way up there, you have the organism. So this is a great way to visualize the complexity of humans and how the information can trickle from the genome all the way to the phenome. But we can get derailed at any point, and the information can get diluted very easily. And so this is a very complex space. And just to illustrate how complex it is, this is just a metabolic map. It's just one of those layers. How can we possibly, as humans, integrate all of this information and come out with a response that is always accurate and reproducible? So we can't. So this is what computers can do a lot better than we do. So I like this image on the left because it shows how we can represent different variables from what we call the endome. So from the skin in, all the variables that are molecular, cellular, immunophenotyping measurements, and those from the ectome, from the skin out. So exposures and diet and so on. And then we get the opportunity to work together with Sylvain and more recently with Einar. How can we translate this into a model in which we use this integration, but now to create Earth analogs of what it might be space travel? So now the variables that we want to integrate are those that occur on Earth, and how can we learn in order to infer certain behaviors of things that we cannot infer. Unfortunately, the data collected in space is very limited, but perhaps if we find the right analogs, then we might inform ways to develop countermeasures for things that we want to avoid in space. So this is the biomedical knowledge graph, spoke. This is a metagraph. Each node here represents a type of concepts. We have genes, we have proteins, we have pathways, we have compounds, metabolic or pharmacological. We have certain different levels of granularity, microRNAs, we have protein domains. We also have gene ontology, of course. There's 11 different ontologies integrated into spoke. We also have organisms that are of importance to human health. So although spoke is human-centric, of course, we coexist with the millions of microbes and viruses that are part of our landscape. So we include several thousands of those. And then we also have, of course, diseases, symptoms, side effects, and so on. So spoke contains the information of about 55 databases right now, about 35 to 40 million concepts related by close to 150 million relationships that are semantically determined. So this is just a few visualizations of Spoke. You can visualize all diseases at once if you want. This is the disease ontology. You can zoom in and out and find all the cancers together, all the immunological diseases. Actually, they're colored by that now. But then once you find the disease or the diseases that you're interested in, you can jump to, okay, give me all the neighbors of this disease in gene space. So all the genetic information about these diseases, or give me just the symptoms, or give me just the therapeutic approaches that are used to treat this disease. Here's the bacterial taxonomy. We have more than 350,000 different strains of bacteria, each of them with their encoded genes. And then we have their proteins that they encode, and then we have the interactions between those proteins and human proteins. And so you can see how everything is connected. Metabolic pathways, this is a hierarchy of all metabolic pathways, highlighting the central carbohydrate metabolism, and then the glycolysis pathway, and then you can zoom in and you can see exactly all the steps from glucose to pyruvate and so on. So at each of the steps, again, you can go into a different direction, ask a different question, which are all the reactions that produce this particular metabolite, or which bacteria encodes the machinery to process this particular metabolite, and so on. A little bit about the ectome. So in the ectome, we incorporate, again, exposures or population level information. So prevalence of diseases by city or county level. That depends on how the information is exposed. But then we also download databases from the Environmental Protection Agency. So contaminations, both in soil and water and air. So that's also by county level or zip code, even in the United States. So we can represent the abnormal levels of certain chemicals or pollutants in each zip code. And why is this important? Well, for medicine it is, because sometimes it is this determinants that are sometimes equally or more important than what we can measure from the patient. Talking about other determinants, social determinants of health, they're incredibly important too, which have been largely neglected because they're difficult to measure. But now there's a lot of efforts trying to, first of all, create an ontology of them, which is very difficult. Maybe LLMs can help. And then to record accurately some of these by surveys. And so the NIH is doing different surveys, and these are very, very large in terms of scale, and we collect that data and put it in by county level. So then with all of this at hand, then we can ask questions. For example, how can we move into a direction where we can create a model that not only is statistically valid, but also understands biology? So going towards the explainable part of the explainable AI. So in this regard, what we did is we developed a series of algorithms that allow us to merge or to embed individual patient data on the left-hand side of that figure with Spoke. Spoke does not contain patient data. Spoke contains basic information, boring textbook information that you will find anywhere. But then you may have a patient that you may be interested in relating those variables. And then we can embed this patient using the EHR information onto Spoke. And then we can create a Spoke-based representation of that patient, which is this, what we call a health barcode or a Spoke signature that represents the state of that patient at that particular point in time, or an aggregate of a certain time window. And then we can do the same with the next patient, and with the next patient, and with the next patient. So this is an example of how we use that approach to create a model, a machine learning model that can identify individuals who we know will be developing multiple sclerosis in some time in the future, but we're not telling that information to the model, which is grabbing the data 1 year, up until 1 year before the diagnosis, the first diagnosis code was entered in the record, or 3 years before, or 5 years before. How far back can we go and still detect those combinations of variables that might be predicted? And in this case, the model did quite well, even at 3 years before the first diagnosis code was entered. I've got an area under the curve of 83%. Of course, the model, the performance of the model gets better as time to diagnosis approach, because there's more information trickling down through the network as time to diagnosis approach. And we can see that very clearly with this example in which we're doing the same, now predicting Parkinson's diagnosis. So we did the same type of selection of data. On the top are the individuals who will develop Parkinson's at time 0, and on the bottom, individuals who never develop Parkinson's. And then we're comparing them at different windows, different time windows. And here, what I'm going into is that this is explainable. So it's not a black box model that tells me, yes, this is the prediction, but I don't know how. You can track node by node in Spoke how much information was contributed by each node. And in this case, the Parkinson's disease node itself, that's the node of that represents the disease in Spoke itself, you see that it acquires more and more importance as time to diagnosis approaches. That red line there is the significance of the, when we compare the significance of that node between patients and no patients. PD, individuals who will develop PD versus individuals who don't. So as you can see, that node becomes more and more important as time to diagnosis approach. The same for gait apraxia, so abnormalities in the gait or tremor. In contrast, when we look at, as a benchmark, when we look at concepts that have nothing to do with Parkinson's disease, they are not any more important in the individuals who will develop Parkinson's compared to those who won't, as time approaches the diagnosis. So that kind of tells us that the context in graph space is relevant for the diagnosis, for the prediction of diagnosis. And then when we go to the performance of the classifier, in this case, again, as time to diagnosis approaches, the model performs better. Of course, the gold standard is, well, how does the neurologist do? If we give the same information that we train this model to a movement specialist neurologist, what will be the comparison? Well, it's a little bit unfair, but the model did better than the neurologist in this case. It was a specialist in Parkinson's disease. We told him, we're going to give you 100 cases. We're going to give you all the variables that we trained the model with, and you have to tell us which of those individuals develop Parkinson's in 3 years. Of course, the neurologists will have more information that we couldn't embed in Spoke at that time, but with the same type of information, the model did better. So, which is a lot to say because you can imagine that a lot of the symptoms and abnormal labs that these individuals may develop might be too unspecific to raise the level of awareness to say, okay, yeah, this might be something going on. But deep down in the network, some of those variables might connect, and then the emergence of that diagnosis may be more obvious for the model. So how does this relate to space? This is where Einar came in with the concept of, well, maybe we can use this to inform the way in which we're utilizing medicine for space flight in terms of finding analogs on Earth that then we can identify in those variables that are relevant for space flight and try to mitigate them. And this is also where we came together with Sylvain and got interested in GeneLab, because GeneLab, as Walter described earlier, contains thousands of experiments that were performed both at the ISS and on Earth, but with relevant variables. And what Sylvain was interested in is, can you use Spoke to infer higher order phenotypes than the ones that we can measure from these mice? So in this particular example, we analyzed a few missions in which mice were flown into the ISS for 30 days, and then they were compared. The gene expression profiles of 3 different organs were compared between mice who were 30 days in space versus time 0 or Earth. And of course, all the RNA-seq data was performed. It was analyzed by GeneLab, and then they identify certain genes that went up and down in each of the 3 organs, and that was great. But then what else can we find with Spoke? Can we infer some higher order phenotypes in these mice? And the short-term answer is yes. This is, again, a much more simplified version of the metagraph of Spoke. But if we look at the top 10 pathways that came out of this, you recognize some of them, and it makes a lot of sense. DNA damage response, or defensins, and toll-like receptors, innate immune response. Those are the ones that they had also identified by regular pathway analysis. But then if we look at the top 10 symptoms, for example, well, we predicted that these mice will be, according to the gene expression profiles, they might be feeling fever or pain, eye manifestations. And we look at some of the top side effects that were scored: nausea, rash, dizziness, headache. So these were symptoms that were reported by astronauts going into this International Space Station. So this was something that gave us indication that it might be a useful thing to infer different phenotypes from the same molecular data. So this is where we engaged formally. Now we're in a NSF-funded project with Sylvain and Einar, in which we're utilizing both data experiments from GeneLab and social determinants of health and other types of information that we can get on Earth to try to integrate and create analogs on Earth. So this is just a snapshot of some of the experiments in GeneLab. And what we're doing is, first of all, we selected which were the data sets. Taking time. Okay, 5 minutes. So we checked of all the almost 450 RNA-seq experiments that are there, how many we can process. Some of them were already processed. Some of them were in organisms that Spoke does not understand or does not care for now, like plants and C. elegans and so on. But then we came up with a final set of experiments that we were ready to integrate. Likewise, we did something similar with large surveys of social determinants of health that we could integrate into Spoke. And the way that we're doing is we're creating sort of the concept of a Spoke fabric. And the Spoke fabric is without getting into too much technicalities, is an additional graph that will act as an interface between the experiment, the raw data itself, and Spoke. So it's a graph that understands the structure, the metadata of the experiment, and that can transform it into something that Spoke can understand. So we developed a Spoke GeneLab fabric, and now we're working on the Spoke social determinants of health fabric, and we have additional fabrics in the making. So as one example of the social determinant of health that we're working on is, for example, social isolation. So as an analog, naturally, we go to surveys that will, first of all, have information about which are the concepts that may signify that someone is suffering from social isolation, and we identify those as MESH or SNOMED concepts. And then we can look into our UCSF wide clinical data warehouse that we have more than 6 million de-identified electronic health records from patients, and then extract the cohort of patients with that particular set of characteristics. And then we can embed those cohorts into Spoke and identify the analogs that we would like then to infer and to make the most similar to what an astronaut might be feeling in space. So this is the way that the Spoke fabric for social determinants might work. In this case, I think we're putting in the prevalence, and we have them by, in some cases, by statewide or by county or zip code. So there's probably more detail than I wanted to share, but the idea in the end is to, again, use both ends of the spectrum, the molecular and biological side, and the social aspect. So to see whether we can create, again, those analogs utilizing an integrator tool like Spoke. And in the last 3 minutes, I put this together right after hearing Walter, because again, LLMs are everywhere now, and how can we capitalize on that? And Walter spoke about RAG. So we implemented RAG for Spoke, which we call KG RAG, so knowledge graph RAG. And the concept of KG RAG here is we use Spoke as a context source for any prompt. So once the user sends a prompt, then the first thing that happens is entity recognition. Is the prompt does the prompt contain any disease or gene or symptom or something that Spoke understands? If it does, then it goes to Spoke, it retrieves the neighborhood of that, and then it injects it back into the answer. And this is now in press in bioinformatics. And we also implemented a chat version of that. So I don't know if this will work, but I'll try. So.
Hello, I'm Spoke, the scalable precision medicine open knowledge engine. How can I assist you today?
So you can ask a medical question or a medical question.
The genes associated with phenylketonuria are PAH, SPR, QDPR, PTS,
and it tells you the provenance. ChatGPT won't do that. It tells you where this comes from. And then you continue and you can go deeper and deeper. And then that lag that you see processing the info is checking.
Phenylalanine 4-hydroxylase, PAH, provenance uniprot. Yes, the protein encoded by the gene PAH, phenylalanine 4-hydroxylase, PAH, is an enzyme, provenance uniprot.
So the idea, again, you can go deeper and tell me all the reactions that this enzyme catalyzes and what are the reactants, what are the products, and so on. So we're now working with the Versa team at UCSF to enable anyone who's using Versa to deploy this little drop-down menu here. Use Spoke for my answer. If I have a biomedical question that requires any genetics or biochemistry or physiology or symptomatology or anything that might be informed by Spoke, we can request that version of Versa. So we're deploying it in the next couple of weeks. So with this, I know it's time, so stay tuned. I'm sure that there will be more to share with this same group in the next iteration. But for now, it's been a great pleasure to present to you, and I'll be happy to take any questions. So this is all the people who participate. So.
Great, Sergio. That was really, really interesting. If you could stay up here, and I need to introduce another person who's helped me a lot, and that's our UC Space Health intern, James Rohde. And James, if you don't mind bringing the chairs up, we're going to have both of our speakers up here so you can ask each other questions, or the audience can ask questions. There is a lingering question from Zoom while we're getting set up for this, and I think it could go to both of you. This person in particular was interested in any experiences of AI for analyzing data related to genetic eye disease. So I think if it's all right, we'll have both of you field that question.
Yes, of course, I'm interested in genetics, and one of the first things that we took interest is how can we use this to interpret genomes? And so not only eye disease, and we haven't particularly done that, but the system is ready to incorporate any type of genetic information. So if you have genome sequencing, you can enter a number of variants of unknown significance or known significance, or if you have a genotyping array, you can enter spoke through variants. Spoke contains more than 1.2 million SNPs. So the answer is yes, it's able to. We haven't done it, and we are looking forward to collaborating with people who might want to do some of this work.
Yeah, did you want to answer that? Is there work being done at NASA?
I'm sorry, can you repeat the question?
It was using AI in any of your models to look at genetic underpinnings of eye disease, I think is what the question was.
Definitely. I don't know if this is working. Oh, yeah, we definitely are. Not really familiar with a lot of them, but I know definitely, we worked with Spoke, and a lot of that is being done in our side as well too. So
great. I'm gonna ask James to do me one more favor. Can we take one stool away and I'll just stand up? And so I didn't clarify, but James is a UC Berkeley student. He's our UC Space Health intern. He's already sent research to space, and he has another project getting ready to go up on the ISS. So I'm highlighting that because I want people to know that we have people of all different levels who come to the meeting and interact, and actually we've already made some relationships that didn't exist before. So keep talking to each other while you're here, and then follow up afterwards. And James, if you don't mind at some point letting people know how to reach you, because he's also done internships at NASA, including with Gene Labs, and some of the work that was talked about today, James worked on. So if you're interested in any intersections, here's a product of several. Did you have a question, James? Yeah, great. Since I have totally put you on the spot.
Thank you for your presentations today. I just had a question. So as like a student, I know AI has been really prevalent in the education of like students, and also like in the future especially. So I guess this can go to both of you as like current educators and future educators. What do you think is the role of AI in like medical education in particular? Because like Dr. Bernstein, you said that your model like performed better than a clinician, and I think that's like very amazing. That's like wow. Because so like how do you I guess, well, yeah, like what's your opinion on like the role of AI in like medical education?
Well, I get asked this question a lot, and I gave it a lot of thought, but they always come up with an analogy. Modern airplanes can take off, cruise, find a destination, and land without human intervention today. There's still always two pilots in the cockpit. So I think that the doctors will always be there. AI will help them find the right paths when diagnosis is more difficult. If the diagnosis is easy, they probably won't need it. But when the diagnosis is difficult, or when the intervention might be more difficult, then AI is there to help them don't make mistakes, and to remember things. People make mistakes, people forget things. AI doesn't, hopefully.
Yeah. I mean, my analogy is a calculator. Like, when the calculator came out, some people knew how to use it, some people didn't, but it helped and it aided, and now everyone knows how to use a calculator. And in biology, I think AI has had a lot of usefulness out of it. When I was in grad school, a lot of the models that I was developing were to like bridge like physics and biology together. And I think the data that we get from biology, and the physics that we get, and the models that we design, or that we study, AI sort of bridges that gap and gives you insights that you would have necessarily have found had you had not used a certain tool. So
I think we have a variety of people in the room. Are there other clinicians in here besides myself? Yep, I see a couple. So I might ask you to answer how you see this integrating with your work. I personally see it as augmenting and enabling us and making us more efficient, and hopefully that's the case. And also then creating greater access to more people for quality information. So I don't see it as a threat to replacing, but I do think for all of our fields, it'll be augmenting. Did you want to speak more?
Yeah, that's sure. This will be helpful in terms of cutting time, maybe cutting costs in terms of choosing what is the most performing exam or test for having a diagnosis. And hopefully we can merge it with clinical experience too. So many of these systems still lack some clinical derived data. So there's work to do for it.
I also have been working on how are we going to help astronauts stay safe as we move away from a tethered model. So moving away from the ISS back out to longer missions. So cislunar and further missions out to deep space. So when we do that, we're going to get more and more untethered from Earth. And I think about that in healthcare, we actually need to try to reach more people, and many people can't access centralized healthcare systems. And so as we look at distributed healthcare models, this type of technology and use of informatics will allow us not only to have more distributed care, but also will allow for closed loop processes. So as you can imagine, if you're out in deep space and you're doing an ultrasound, you can't rely on mission control to interpret the ultrasound. And AI is already being used in ultrasound, not just for the analysis, because it's very operator dependent and analyzer dependent, but also being used as a QI quality assurance tool so that you know you're getting and acquiring the right study at the moment you're getting it. So I think there are many, many ways this is going to help us with accuracy, but it's also efficiency, but also distributing care further than we're currently able to do in a high quality way. All right, I took advantage of the microphone. Let's take more questions. Any questions on Zoom, Isaac? Okay, good. I also want to point out Isaac and thank him. He's from our tech support group here at UCSF and done a great job making this look seamless. Are they going to feed those to me by text? Because we have a question in the room we can start with.
Thank you. Great talk, both of you. My name is Fatih Karouya, Blue Marble Space at NASA Ames Research Center. First of all, I want to thank you for accepting the invitation a few years ago to have spoke as part of the Space Life Science special issue that I had within Life Journal. So I have a question for you. Actually, it's kind of twofold. First one is kind of related to the fact that Spoke integrates many different type of data bases, and I was wondering how would you in some way tailor the system to kind of update itself with new type of information from biological as well as clinical side. And then the other one is more related to space and astronauts' health in particular. And then I was wondering how you consider how Spoke can be used to tailor countermeasure for individual astronauts based on clinical as well as genetic information.
Yeah, so first, the updating aspect of Spoke. So the way that Spoke is built now, we write scripts to download the information from each database periodically, almost on a weekly basis, and the graph is reassembled every week. So the data that all the databases that are part of Spoke are current as of this week. If your question was how else can alone bring additional information, we're not there yet. We spend a lot of time vetting which database is worth including, for what reason we want this database, and is it a trustable source? Is it going to be there next year? So there's a lot of human decisions that go into whether we decide to put in one database or not. Maybe in the future there's some other federated approach that can yield something of equal quality and usefulness, but I'm not aware at this point in time. The other question is on whether we can enter, you mentioned genetic data and pharmacological data to develop countermeasures. Yes, I think that's one of the ideas, is to try to build a model that on which we can predict. So if let's say this particular gene expression profile obtained from mice results, we predict that it will result in jet lag or headaches, what might be a drug that we can use to countermeasure that? And if astronauts are in space and they do not have the luxury of talking to a doctor, then they will have an indication to say, well, this might act as a countermeasure for the current symptom. So again, infancy, baby steps at the beginning, but the goal is to create those analogs that can be automated and that can be somewhat intelligent in the sense of integrating more information that we can do here on Earth.
Great. We have a couple questions from Zoom, and we'll start back with Dr. Costas again. So Van Costas, if we can get him teed up.
Sorry, I was just typing my question, but I wanted to follow up on Fatih's question. And should we rethink the way we structure our databases? Can we move towards an embedding that is using knowledge graph instead of the traditional relational data or document stores, that kind of things? Because maybe we can have a better way to handle semantic searches and have better AI applications. So right now we are committed to the traditional database, but I'm wondering if we should entirely rethink the approach in the future to really benefit from knowledge graph.
Well, yes. I mean, I think you're thinking the next level up in terms of abstraction, and I think it's one way. It's just to have something that is so smart that can do that integration without us specifying what the information is and where the information can be found. Again, I think that the answer to that has to do with probably smart federated searches. You can call them knowledge graphs, but it could be anything. I think there's a big component, there's a big human component still needed. And there are automated ways to create knowledge graphs from data in other realms, like in engineering or in e-commerce or a client database. In biomedicine, you still need the human. And I don't know, maybe with better standards and better semantic interpretation of the existing data, we can get there. But I still think that the human is needed to make those decisions, because biology and biomedicine, unlike all the other fields that I mentioned, it's complex. It's not complicated. The tax code is complicated. Biomedicine is complex. So we don't understand yet the rules. We don't understand. So I think there's still a lot of human input needed for that.
Keep thinking in the room here. We do have some chat questions, so I'll try to cover this. I'm not sure I'm capturing it exactly, but one is, how do you ensure that the predictions made by Spoke or the systems or similar, Walter, what you're working on, that first of all, that they're clinically actionable, but also that they can be used in a clinical context? How are you going to create some assurance around that.
I can start. I think that, first of all, it starts with the question that you ask. If you ask a question that the answer might be clinically actionable, then it's worth asking. There are questions that are not worth asking because they're not actionable, and you can get an answer, but is not realistic. So I think a lot of that has to do with choosing the cases that we want the system to be able to solve. We can go into deeper conversation, but I think that's in a nutshell what we have in mind, at least when we train these models.
Yeah, I think in a more general case, I think there are tools being developed, and it is like an active field that's being researched. The tools are new. Tools to measure these tools are new, like I was mentioning earlier. But they, as they're being developed, they start measuring things like biasing, start to measure like things like, so on, like a bunch of different things that I can't remember right now. But I think being able to leverage these new tools and these new metrics, you can then start to compare a lot of these different models. There's more than just ChatGPT. There's a lot more than just one algorithm out there. And there are different ways and approaches to sort of benchmark these things. And so I think as the field matures, I think we'll have a better insights into how to like best be able to apply these into like not just in clinician settings, but also in space flight. That's also like an area of research that we're also working in. One of the things that we're doing right now is that we're testing these large language models and implementing knowledge graphs to see how well they perform and the types of answers they're going to give you. Because when you're on a mission to Mars for 2 years, you're not going to have access to some of these resources. And so having a model there and having the data set in such a way that the large language model can give you an answer is going to be pretty crucial. And so by testing those models here now, we can start developing some of those benchmarking tools. So
there's also a value in the ability to do much more multi-parametric risk prediction. We're starting to send a wider substrate of humans up to space and into that very high risk environment and far away. So as we look at that, we have highly selected and highly trained people that's the NASA astronaut pool, but we're sending more and more people now from age 20 to 90 with all types of comorbidities. So the risk prediction side of this type of work, I think, is something that's going to be very important as we go forward for the longer missions, but also as different types of humans go. But then we can translate that so that as people come into our ERs, we're going more and more towards precision medicine. We're working at an individual level of what's their risk, what's their exposure in their ectome or their exposome, et cetera. And then what might be the most targeted, least toxic therapeutic for them. That question did come up in how you see these working into precision medicine. And on a practical level, one question was, if you're getting data from the EHR, like we talked about social determinants of health, and integrating that into Spoke, do you ever see Spoke as a tool that's embedded in the EHR?
Well, I'd rather see the opposite as implementation wise, but we have an example in which we would love to expand that we developed an API to go from the EHR to Spoke and back. So essentially it's like a KG RAG, but instead of the knowledge graph informing the large language model, the knowledge graph informs the physician directly. So physician is in front of a patient, has variables, can click a button and select a set of symptoms, labs, and medications, comorbidities, and then send to Spoke. And then Spoke will come up with, okay, these are the 10 most likely conditions. These are the 10 most likely side effects. These are the 10 most likely. So that's something we're testing now in one clinic. We'll love to expand this. There's some technical things and legal, ethical things to navigate. But the idea is absolutely is to augment the information that a physician gets in real time.
I think that's what they meant, some sort of integration. So that was very helpful. And Walter, I know that you're directly working with NASA. Are you doing any work with SANS? That's still the space associated neuroocular syndrome. That's still one that is, it's very unclear who's going to get it, who's going to be most affected by it, et cetera. Is that any work that's going on with either of you?
No, I'm not working on it. But I know that there are definitely other researchers in the space bio research branch that are. And so, but yeah.
Those things like SANS where it's just, we know it's prevalent, but we don't know to what extent and who's at most risk. Same thing with bone loss. We actually, so instead of treating one size fits all with these types of models, I think we're going to get more and more to the precision or individualized approach. We have a question in the audience in the room.
Thank you very much. And thank you for the organizer, Einar, and the presenters. My name is Dan Lappin, and I'm an independent researcher, and I work in the arts and humanities side of astrobiology and exobiology, looking at potential applications of early cosmochemistry and cell structures into pediatrics and family medicine, how we understand our models of anatomy and physiology from a human perspective. Okay, so here's a philosophical question. About 40 years ago, there was a book by an author named John Nesbitt called Megatrends. And one of the themes out of that book, of the top 10 trends in society 40 years ago, was the concept of high tech and high touch. The more we go towards high technology, the more we're going to realize that we need humans. Okay? And so the question philosophically is, what is the domain where we absolutely need a human? Where we don't have a model, we don't have a foundation, when we have this extreme mystery like origin of life. Okay? And we're asking questions that haven't been asked before. We're getting an astromaterial sample off of Bennu asteroid, something like that. Philosophically, then what arises? How can we ask questions into this mystery? The more we go to high tech, is it inviting us to ask new questions differently and ask questions differently into the mystery?
Oh, yeah, I love the challenge. Certainly, there's always going to be a place for human interaction. I don't think I don't really subscribe to an apocalyptic vision of the future where we're taking over. I think the humans will always have a place. And I've also read books in which the prediction is, well, depending on how humans treat AI now, AI will treat humans later. And if we're smart enough, they will never overtake our civilization. So I think these are valid points, but the human is always going to be part, and there will be things that will be solved by AI that humans cannot. I mean, as we keep doing research and keep improving the number of computations per second that our computers can do, we can get to more detailed answers of things that we never thought before could be possible. So now we can predict the protein folding and the activity of protein space and sequence. And 20 years ago, that was a dream. Maybe in 20 years, we can identify how consciousness emerges from neuronal inputs and activity. I don't know. But I think there's still that perception that humans will be out of the loop. I personally don't think that will be the case.
Walter, do you want to comment on that?
I will try. I'm of the belief that AI is a tool, and this tool, I think, works really well for us. And what I'm realizing just on a day-to-day basis is that I can get a lot more done. And it's because a lot of these things are sort of encoded into the training set that these large language models use. And for me, I've always just seen it as a way to sort of allow me to ask questions and find answers in a sort of more efficient way. One of the projects I got to work on was, applying machine learning to try to determine whether the samples that were these lipids that we're finding in Mars, whether they're abiotic or biotic. And I think these tools allow us to sort of probe those answers, extract a lot of data, and then find patterns that we might have missed. And so To me, I think sort of at the core of all this is data, and data is generated by humans. And so I think that's when we use that data and we train these models on that data, I think we get a really useful tool that allows us to, ask questions and find patterns that we might have missed. So
Thank you. Those are both very insightful and prompted by a really interesting question. We have one more question from chat, and this is from Ryan Scott. If anybody knows, he's deeply a part of this team with Costas and also with Sylvain Costas, and also has been very helpful to me in the work we do in UC Space Health. So have we got Ryan up there? All right.
Hello, hello. Is the audio working? Oh, wonderful.
Yes.
Hi, Sergio.
Yes, you're on
Hi,
the big
Walter.
screen.
I just wanted to give a pitch to the SANS question that came up. That's the Space Adaptation Neuroocular Syndrome. There are two current analysis working group projects that are mining retinal data and other, like I think intraocular pressure, and a couple. These are from rodents. I wish we had human data, but there are two open access projects if anyone would like to join them. One is looking at Rodent Research 18 data. The other one's looking at Rodent Research 9 data. Some of it is reduced by sulfite sequencing data. Some of it's also RNA-seq, bulk RNA-seq. And if anyone's interested, these are open access projects of data that are, if people would like to join in and find some cool insights and contribute to the exploration of humanity throughout the solar system and the stars. So, yeah, just wanted to, the SANS question came up. So just wanted to plug that because it's,
Ryan,
and then eventually
thank you.
Spoke will be great to be able to be another tool in the toolbox for people. So excellent.
Thank you for that and for sending us off on such a open, welcome invitation to collaborate. And the hybrid vigor that comes out of this is obvious, I think, just listening to these two and the work we all do together. I think what I'd like to do now is thank everyone in the room for coming, because you actually create what really is a rich environment for discussion. And we're going to be doing these monthly. And so please stay tuned. You're on the list now. If you signed up for this, you'll get notified of the next session. And if anyone wants to speak from the UC side or the NASA side, or you have suggestions for speakers or topics, please let us know. These QR codes relate to the different organizations here that are involved in this. But since you're now signed up, you'll get notifications, but feel free to invite other people and let us know what topics you're interested in. Thank you again for everyone who made this such an interesting discussion tonight. Thank you very much to our speakers.